mentorship of Louis Ganem and Yusuf Barash. And I'm really excited to share with you guys today a story that we've been working on for the better part of the last year about G-Quadruplexes in untranslated regions of UTRs. So broadly, what's the problem? The problem is, as the cost of whole genome sequencing has fallen precipitously over the past 10 years or so, and the number of individuals who are receiving whole genome sequencing has increased significantly, our ability to interpret and understand the impact of genetic variation, particularly as it affects non-coding regions of the genome, has lagged behind. So on average, an individual is estimated to differ from the human reference genome by approximately 5 million genetic variations. And most of this variation affects non-coding regions of the genome, where hypothesized regulatory elements interact to produce differences in biology that we can observe between different tissue contexts, between different people, and between disease and healthy states. Understanding the impact of variation affecting these non-coding regions of the genome is therefore super important for understanding biology and disease. But this is problematic because of two primary reasons. One, we don't know where all of the regulatory elements of the non-coding genome lie. And two, when a variant affects a non-coding regulatory element, we're not sure exactly what the potential impact of that variation is. So we're particularly interested in understanding what G-quadruplexes do within the transcriptome. So G4s are stable secondary structures that are composed of stacked quadruplex guanine repeats. And a canonical G4 sequence consists of trinucleotide guanines, or more, separated by 1 to 7 other nucleotides, repeated 4 times. So it's known that in vitro, these sequences will fold to form very stable secondary structures that are mediated by non-canonical hydrogen bonding between the guanine nucleotides. These structures were first described in telomeres of DNA, where they're hypothesized to protect the ends of DNA from degradation by cellular factors, and relatively less is known about their biological function and importance in RNA. We do know that in RNA, however, these G4s tend to form parallel strand structures where the 5 to 3' ends of the strands are all aligned in the same direction. So we became particularly interested in G4s within UTRs because of a study that was published in 2015 that performed transcriptome-wide structure mapping of putative G-quadruplex forming sequences. In this study, they were able to show that there was actually a particular enrichment for G4 structures in the 5' and 3' UTR of protein-coding messenger RNAs, suggesting that perhaps these structures are playing some kind of regulatory role. About a year later, however, a second group published a paper describing that most of these secondary structures, when looked at within the cell or in vivo, were actually unfolded in eukaryotic cells at steady state, suggesting that perhaps the secondary structure is actually not important for their regulatory function, and leading some to believe that perhaps RNA G4s and UTRs are actually not functional at all. Our hypothesis was that because these RNA G4s were enriched within UTRs, that they could be regulatory features. And towards asking this question, we asked two smaller questions: whether or not there was evidence of heightened selective pressures acting over these UTR G4s, and whether or not there were evidence of enrichment for functional associations or variations within these G4s that were associated with changes in gene expression or perhaps disease pathology. And in order to explain our approach on assessing the degree of selection acting over these G4s, it's useful to go back to an example that was made famous by Abraham Wald, who is a statistician working with the US Military Research Group during World War II. So the problem that Wald was presented with was, given a certain distribution of damage that accumulated on airplanes as they were flying their bombing missions and returning to base, where can we distribute armor on these airplanes so that they would be more effectively made resistant to being shot down? The prevailing thinking at the time was where there was higher concentrations of damage on these airplanes was where we needed to reinforce these airplanes the most. But Wald realized that patterns of damage on these airplanes that were actually only observable when the airplanes were not destroyed. So patterns of observed damage on these airplanes actually represented regions of these planes that were more tolerant to damage. In contrast, regions that are depleted of observed variation or damage on these airplanes were more critical components of the airplanes, which needed more reinforcement. We can take the same approach to the analysis of human genomes at population scale by recognizing that because negative selection is continuously removing deleterious alleles from the population, regions that are depleted of variation within human genomes are likely more functional. And we can do this using resources such as Nomad, which contain over 140,000 whole exome or whole genome sequences. And in fact, this was something that they did in the original EXAC and Nomad studies, was look at the rate of variation across protein coding genes to produce a ranked list of the most constrained protein genes to the least constrained protein coding genes. In our case, we can assess sequence constraint by 3 metrics: whether or not we observe a reduction in allele frequencies for variation affecting UTR G4s, whether or not there's a depletion of polymorphic sites, and whether or not we see enrichment for rare variation. So to address the question of allele frequency within UTR G4s, we first mapped the canonical G4 forming sequence transcriptome-wide using Ensemble within UTRs. And we produced 2 sets of G-Quadruplex. One is an RG4-seq validated set of G4s. So these are G4s with in vitro experimental evidence of secondary structure formation. And the second is a set of putative G-Quadruplex. So any sequences that match the canonical G-Quadruplex forming motif, we call the putative G-Quadruplex. Our null hypothesis is because there's no difference in the strength of selection acting over these regions, we should not observe a difference in allele frequencies across G4 versus non-G4 forming regions of UTRs. So we take our mapped G4s, and then we additionally match our G4 versus non-G4 UTRs by the degree of protein coding constraint acting on their protein coding genes. I'm not going to go into specific details of how we do this, but we did this because we did not want linked selection from highly constrained protein coding genes to be affecting our estimate of allele frequencies within these UTRs. So we look across these contexts of G4 versus non-G4, we see that there is a significant reduction in allele frequencies for variants that affect G4s versus non-G4s, looking specifically at the G tracts of the genes. We next turn to ask whether or not there was a depletion of polymorphic sites that would be consistent with the activity of negative selection. So because it's known that within non-coding regions of the genome, the probability of observing a mutation is highly dependent on the local sequence context of that mutation, we had to control for this local sequence context by applying a mutation model. So our colleagues at the University of Pennsylvania had developed a model that was able to accurately predict or explain 81% of the variance in nucleotide substitution probabilities based on a 7-mer heptitide context within non-coding regions of the genome. So we took this model and we applied it to UTRs within G4 forming regions versus without G4 forming regions. With this model, we can compare the observed number of polymorphic sites against the expected number of polymorphic sites to see how much G4s would or would not deviate from the null model. So we applied this to non-G4 forming regions of UTRs first. As denoted by the red bands here, we see there's relatively good fit between the observed number of polymorphic sites versus the expected number of polymorphic sites for both 5' and 3' UTR contexts. When we use this to look at the rate of observed versus expected polymorphisms within G4s, however, we see there's a significant depletion of polymorphic sites within the G tracts specifically. So the N1 to 7 linker nucleotides don't seem to be under any stronger selection against polymorphism compared to the G tracts, which are potentially important for secondary structure formation. We finally can ask for whether or not there's an enrichment for rare variation. And in order to do this, we apply a mutability-adjusted proportion of singletons model. So proportion of singletons, a singleton is a 1 variant observed in all of Nomad. Proportion of singletons based on the mutability of the local sequence context. So we take into account the local sequence context, and we calculate the expected number of singletons we would expect to see given that local sequence context. This was actually done first in the ECCB paper, where they attempted to quantify the degree of negative selection acting over different classes of variation, genome-wide, where they correctly saw that there's in fact stronger negative selection acting on essential splice sites or nonsense sites. And they noticed that actually, when you look at all of 3' UTR or 5' UTR variants as a whole, there doesn't seem to be any stronger negative selection acting over these variant classes compared to synonymous variations within protein coding regions of the genome. When we apply this to UTR G-Quadruplex, looking at the trinucleotide G tracts, we see that compared to synonymous, missense, and putative loss of function mutations, variation affecting the second G of this G tract tends to be significantly more enriched for rare variation compared to synonymous variation within protein coding regions of the genome. And this is consistent across 3 different classes of genes we looked at. First are all genes. The second are the top 3rd most constrained genes by their protein coding estimate of degree of constraint. And the final is a set of disease genes which have been associated with some kind of disease pathology in ClinVar. And in some cases, variation affecting the second G of the G4 G tract can be comparable to even missense variation. When we look at non-G4 forming G tracts, so any trinucleotide G or C context within UTRs, we don't see a significant departure in the enrichment for rare variation, suggesting that perhaps this enrichment for rare variation is specific to Gs that are involved in G-quadruplex formation. So I've shown you that allele frequencies and the number of polymorphic sites are reduced within G-quadruplex forming regions of UTRs. I've also shown you that rare variation is enriched within the second G of the G-quadruplex, which coincidentally has been shown in some studies to be the most important G for maintaining thermodynamic stability of the structure. Given the evidence for heightened selective constraint acting over UTR G4 regions, we hypothesize that variation within these regions, when it does occur, is likely to be more functional. So we can ask the question of whether or not when a variant occurs within a G-quadruplex forming regions of a UTR, if that variant is more likely to be associated with changes to gene expression, or if that variant is going to be a cis-EQTL. So we can take all of the cis-EQTLs that are mapped in GTEx and ask whether or not we see an enrichment for these cis-EQTLs affecting G-quadruplex forming regions of UTRs. We can do this for different sets of EQTLs. So here I'm showing you on the furthest of your left lead EQTL. So these are the lowest p-value variants that are associated with changes in gene expression for a given gene, or a set of causal EQTLs that are defined by a different study, or nominally significant EQTLs. There's enrichment for variants that are associated with changes in gene expression across 5' and 3' UTR G4 contexts. We can additionally overlap RNA protein binding sites with these G-quadruplex forming regions that are published by ENCODE, and we see that within 3' UTRs specifically, there appears to be an enrichment of cis-EQTLs that are also interacting with RNA binding protein binding sites, suggesting that variation in these regions can modulate changes in gene expression by changing protein-RNA interaction dynamics. When we ask the question of whether or not there's a specific effect of a variant when it affects a G tract, so presumably it's disrupting the stability of this G quadruplex, versus when it falls within the N1 to 7 linker sequences, we see that there's a specific bias for 3' UTR G quadruplex when a variant affects the G to be associated with increases in gene expression, suggesting that the native G quadruplex structure might be associated with maintaining gene expression at a certain level by applying negative pressure to the level of RNA. And finally, because we have all the ENCODE RNA protein binding sites, we can overlap those with G-quadruplex forming regions of UTRs. And we see that when you are in a G-quadruplex within the UTR, you're significantly more likely to be overlapped by an RNA protein binding site compared to if you're not within a G-quadruplex region of the UTR. We can dig a little deeper and look at the specific RNA binding proteins that tend to be enriched for binding near or over RNA G4s. And when we do this analysis, we see that across 5' and 3' UTR contexts, in the different cell lines that are available in ENCODE, certain proteins tend to pop up more frequently than the others, including FASKD2, PRPF4, and GTF2F1. This suggests that these proteins are binding a secondary structure component of these UTRs that is independent of the UTR context alone. I should also additionally mention that when we looked in the literature, we did not find other evidence describing these proteins as G4 interactors. So taken together, the enrichment for cis-EQTLs and RNA protein binding interactions over these UTR G4s suggests that they're variously involved in transcriptional and post-transcriptional regulatory processes. When we look at patterns of shared binding for these RNA binding proteins, we see certain clusters emerge, particularly the one in the lower left corner here. If we perform a Go enrichment analysis for gene binding targets of these clusters, viral process comes up, which is interesting because there have been previous publications describing the enrichment of G4s within viral genomes. We haven't pursued this line of investigation further. Finally, we asked whether or not G4s are enriched for pathogenic or maybe variants of unknown significance in genes that are linked to known pathology. So we take the set of variation that's in ClinVar, and we subset ClinVar by the set of genes that are known to be associated with some kind of pathogenicity. We then look at the frequency that variants of unknown significance, likely pathogenic or pathogenic variation, occurs within the G4 context of these genes, and we find that within all pathogenic associated genes in ClinVar. There's enrichment of pathogen or variation that is of unknown significance or likely pathogenic within 3' UTR context. And this is true, especially when we overlap this further with RNA protein binding sites. Finally, we are particularly interested in one variant affecting the 3' UTR of HSPB7, which is a gene that's been associated with numerous different cardiac pathologies, and in a recent UK Biobank study associated with hypertension. So this is a relatively common variant. It's predicted to disrupt a 3' UTR G-Quadruplex sequence. So because it's common, we can look in GTEx and look in the RNA-seq of GTEx to assess whether or not disrupting this G4 is associated with changes in gene expression. So we take heterozygotes and we map the number of RNA-seq reads that map to the reference versus the alternate allele, which is associated with disruption of the G-quadruplex. And we see that there's actually significant allelic imbalance favoring the expression of the alternate allele, suggesting that perhaps the underlying association between variation in this gene and the effect of hypertension is mediated by changes in gene expression. So in conclusion, the second part of my talk, I've shown you guys that we found enrichment for cis-EQTLs in G4 regions of UTRs. I've shown you we found enrichment for RNA protein binding interactions. Taken together, our results suggest that the disruption of these UTR G4s can be associated with susceptibility to disease and provide further evidence that these UTR G4s are functional elements within RNAs. So I'd like to close by acknowledging and thanking all the members of the Barash and Ghanem labs. Apparently, I couldn't have done this without any of the help and discussions that you guys have had with me over the past year. And like to point out that we have fun sometimes. We go to dinner as a lab. This is a picture of us at a lab dinner last year around Christmas time at a place called Bernie's. And I'd also like to give a warning that if any of you find yourselves at Bernie's in Philadelphia, to not order the salmon cheesesteak. That's a big mistake. So you heard it here first. There are other members of our lab who are presenting posters, and if you guys are interested in learning more about what we do, I'd encourage you guys to go visit them. And thank you for your attention.
Hi, great talk. So regarding the RNA binding protein connection with G4s, I was wondering, is there evidence in the in vitro data in which those were assessed or profiled that, apart from the canonical motif, maybe I'm not sure whether these are G-rich motifs, but if you have analyzed for evidence of those in the in vitro data sets as well, or they could have been confused with artifacts of amplification in those experiments?
Oh, so like are the RNA proteins binding G-rich motifs rather than just G-quadruplex motifs?
Yeah.
So in that analysis, the background set that we used for enrichment of binding was like G-tracts or trinucleotide G-runs of 3 or more. So that was hopefully to avoid or control for the potential bias that could be introduced by only looking at G-rich sequences. But it's possible that they're non-specifically just binding G-rich sequences. And because G4s are enriched for G-rich sequences, they're going to be binding G4s more often than you expect.
Thank you.
Hi.
Hi.
Did you find any relationship to the experimentally validated structure or open G-quads? And what do you think in general about the openness versus closeness of, or structuredness of these things?
Yeah, so in that original 2016 study where they reported that most of these G4s were unfolded within eukaryotic cells, they also saw that when you put a G4 sequence in a prokaryote, it actually stays folded. So one of the explanations for that was perhaps there are proteins that are binding or other factors that are interacting with these secondary structures within eukaryotic cells that are maintaining them in an unfolded state at steady state. I think the question, whether or not they showed that these structures aren't transiently folding or maybe doing something else, remains an open question. But it does appear that by the way that they looked at it, most of these structures are unfolded within eukaryotic cells.
And are the ones that are more constrained more open or more closed?
Yeah, that's not something that we've looked at, but it would be interesting to look at.
So very similarly, there's a very small number of motifs that respect the canonical motif that have been shown by shape to either fold in G4s or not fold in G4s.
Yep.
Did you look at these? And
we haven't, but that would be really interesting to look into. And yeah, so we wanted to keep, we thought that we wanted to try and keep things simpler by following the canonical motif, but it certainly would be interesting to look at non-canonical G structures or sequences that fold into G4s and see if we see a similar pattern of constraint.